Today we will discuss about the reproduction in bacteria. Reproduction in bacteria, right? So as we know that we have two kinds of reproductions. First is sexual reproduction, right? And second we have is asexual reproduction, right? So in sexual reproduction, basically two parents are involved and there's a formation of gametes too. The gametes are also formed, right? But in the asexual reproduction, we have only one parent is required, and there is no formation of gametes, right? No formation of gametes. Now we have basically some types of sexual reproduction too, which like we have meiosis here. Meiosis is done, which is known as recombination. And we have three types of recombination. First is conjugation. Second we have is transduction. Then we have transformation, right? And in this asexual, basically it is known as mitotic division, or mitosis is occurring over here, which is also known as basically we can say this way, some kind we can say it as a mitosis. We have three kinds of this too. Like first is we have binary fusion. Second is we have basically budding, and then we have which is conidia. This binary fusion is known as amitosis, or it is also known as cancerous cell division, right? This is known as cancerous cell division, this binary fusion. So let's discuss about this binary fusion. Basically in binary fusion, this is binary fusion, the cell divides into two equal parts. Cell divides into equal parts, right? So basically let us make, and before the cell, like before the binary fusion, the genetic material copies. The genetic material copies basically in the mesosome, because we know that mesosome is an extension of the cell membrane of the bacteria, in which reproduction, respiration, and these activities are taking place in the mesosome, which is the extension of cell membrane, right? So let's just discuss, like we have a cell over here. This is a bacterial cell, and we know the bacterial cell have a cell wall. This is a genetic material, right? This is the first step. Then what will happen? There will be a replication of the genetic material, like before the cell division. Now what will happen? These two genetic material, like these are the nucleus, basically they will move to their respective poles. But without spindle fiber. Like we know that in humans, when we read mitosis, there are spindle fibers which basically they attract, or you can say they bind to the they bind to the chromosomes in order to attract or to basically attract the chromatids to their respective poles, right? But here we don't have any kind of spindle fiber, so the genetic material move to the respective poles by their own self. After this, what will happen, like this is, there is extension 2, then this cell will divide into its two daughter cell of bacteria, right? This is how the cell division, you can say the binary fission or even fusion, takes place in the bacteria. So I hope this is clear to you. If you have any kind of queries, you can ask me in the comment section and I'll be very glad to answer you guys. Basically here the transverse septum has occurred. You can write over here that here transverse, like we have two sides of bacteria, right? This, if we cut this like this, it is known as lateral, or you can say this is a longitudinal side, right? If we cut the bacteria like this, it is known as transverse side. So here we have the transverse septum has occurred, right here. Oh, in the last step, transverse septum has occurred because bacteria has divided into this transverse section. So I hope this is clear to you.